Hi, I'm Laurel Smith. Today is January 28th, 2002. I'm in the microbiology class, Bio 225, and I am doing my Lab 5, Bacteria, Prophylology and Staining Techniques Experiment and Results video. Here's my paper. And I will be taking the Crystal-Gram Violet and placing it on my cheek cell slide and my plaque slide. Once I drop one drop of the Crystal Violet, I will smear it with another clean slide, and I will pass it over the fire 5 times, and I will get my results through the microscope by observing it. So I'm going to place one drop of the Crystal Violet stain right in the center where I place my cheek cells and my plaque cells for my teeth. I will smear it with a clean slide. And this is what it looks like. I am going to do the same thing with my plaque gums. Okay, and I'm going to let it dry. I am going to place the cap back on the violet stain. While that is drying, I am going to bring my test tubes for my my staph epidermis, my yeast, and my E. coli, where I use them from my first lab, as I had it in my refrigerator as I was told. And I just want to give you my setup of my lab test area. Everything was cleaned, 10% bleach before I started everything. I have my alcohol with my pipette. I have my distilled water with my pipette. And I have my slides of my E. coli, my yeast, my staph epidermis, where I will be placing from my test tubes here to the circle. And then I will be placing my Congo red and my decolorization and also my PVP iodine. And there's my fire to do my sterilization. And this is where I'll be placing my slide, excuse me, for my tests, for my tray staining. Okay. So the first thing I'm going to do is go ahead and grab my staph epidermis test tube and my slide. You can see. I'm going to remove the cap. And I'm going to sterilize it. Okay. I'm going to get my alcohol with my pipette. And I'm just going to grab a little bit of my staph epidermis, and I'm going to place it right on my slide, just one little drop, smear. Okay. And I'm going to place this back into the alcohol to get cleaned and put my top back on my staph epidermis. I'm going to now move on to my yeast to do the same thing. I'm going to sterilize the tube, place it on my yeast Cervera slide. And it is sterile. I'm going to take my pipette out from the alcohol. And just place it in my yeast to get just a little bit. And place it on my slide. Yeast from the test tube is going on the yeast of the slide. I'm going to drop the remaining of the yeast from the pipette back into the yeast of the Cervera. Place the tube back on, clean out the pipette, and do the same thing with my E. coli. Test tube here, slide there. Going to sterilize. Grab the pipette that's been cleaned from alcohol. Get just a little bit of E. coli. Place it on my E. coli slide right in the center where I made the circle. Place the rest of the remaining E. coli back in. Pipette went back into the alcohol. I am going to move this to the side. I'm going to go back to my cheek cells where I place the crystal violet. I have it as my holder. And I am going to swipe it over 5 times. 1, 2, 3, 4, 5. You don't want to keep it completely over the flame for very long because it can break. I'm going to do the same thing to my cheek cells. 5 times. 1, 2, 3, 4, and 5. Okay, we are all done with that. And now I'm going to place with the E. coli. I'm going to smear, place dry. So get my crystal violet again. And I'm going to place 1 drop after I smear. the yeast. Let dry. I'm going to do the same thing with the Staph epidermis. Smear with a clean slide. And the same thing with the E. coli. I'm going to smear. Let it dry. Okay. And I'm going to place the violet ground stain, the crystal violet, excuse me. I'm going to place 2 drops in and let it dry for a minute on all my slides. 2 drops. So that was the yeast cerevera. This is the Staph epidermis, 2 drops on the slides. And my E. coli. Okay. And for my plaque teeth and my cheek cells, I will be looking under the microscope to determine what the total magnification worked best, where I was able to view the cells under the microscope. And I will place pictures there and send them to you. So we are done with the plaque cells and cheek cells. I am going to get a clean slide and I'm going to write the negative, get ready for the negative stain with the Congrow red stainer. And I'm going to write at the top negative MGM on the stain. And I'm going to place the Congo red on this slide. Use streak. And I'm going to use the Congo red and place it on where I did my teeth plaque cells. And smear it. And let dry. And I'm going to do my heat fix times 5 times, and then I'll let it dry for 10 minutes. And then that's when I will look under my microscope to see how that is. And then going back to my test tubes for my, my slides of my yeast cerevera, my Staph epidermis, and my E. coli, I have placed 2 drops of my crystal violet. I'm going to rinse it with distilled water. Here's my yeast. Here's my staining tube. I got the distilled water. Sorry, I'm trying to have it where you can see. And I'm just going to use my pipette just to rinse my slide from the staining, and I'm going to let dry. So I have just rinsed it of the yeast cerevera There is it in the staining cup. I'm going to do the same thing with my Staph epidermis. Going to rinse it. And I'm going to let it dry for a minute for all of them. And I'm gonna do my E. coli. I'm going to rinse it. I'm gonna see if I can bring this over so you can see it. Okay. Going to rinse it with distilled, just tap water. Okay. And I'm gonna let dry for 1 minute. And I'm gonna place my cap back on the Congo Red. And I'm going to do the same thing with the Stafarin. Or no, I'm gonna use the PVP iodine gram stain. Then followed right after will be the decolorization gram stain. And then I will use the gram stain number 4 of the Stafarin. So they have dry. I have my slide of my yeast, 2 drops. Gonna do 2 drops of my Staph epidermis. And I'm gonna do 2 drops of my E. coli on my slide. Let it sit for a minute and I will rinse. Okay, I have my pipette with my distilled water. I have my my dish to rinse. It's been a minute for the yeast severa. I'm going to let it sit for a minute. I'm moving on to my Staph epidermis. And now my E. coli. And it'll sit for a minute. And then we will move over to the decolorizer gram stain. 2 drops of each on each slide. I'm going to move it over so you can actually see me placing the drop on each slide. This has a very strong smell. Even with my mask on, I can smell it. It's pretty strong. Okay, and we will wait 60 minutes to get the to rinse, excuse me. So I will get my tray for staining. And as you can see, that's already the colors of the stains from the other 2 drops. Now we're going to rinse the yeast severa. And the E. coli. And I noticed that on my E. coli, my circle that I printed, that I wrote, is already gone. Okay, last one is the green stain number 4 of the Stafarin. two drops as well on each slide. The yeast severa, the staph epidermis, and the E. coli. So I will bring you to show you that I have put the last drops on my slides. And we will rinse. With distilled water. Okay, and we're gonna let dry for just a minute. And I'm going to do my heat fix on all 3 of my slides 5 times. And then I'm going to view it under the microscope, take pictures, and inform you what was the best viewing of the total magnification on each slide. I have my clamp for my yeast severa, and I'm going to pass it over 5 times. 1, 2, 3, 4, 5, and I will let that sit for 10 minutes. Moving on to my staph epidermis. Same thing, heat fix 5 times each. 5. And my last slide is my E. coli, where we'll do the heat fix 5 times. 1, 2, 3, 4, and 5. I am going to blow out my candle. Well, it won't work with this. The candle has been put out. And in 10 minutes, I will give you my results of what the yeast severa slide looked like under a slide, my staph epidermis, and my E. coli. And again, my name is Laurel Smith. Today is January 28th of the microbiology class, lab 5, bio 225 of the bacteria, microbiology, and staining techniques and experiment of results video. Thank you, and I hope you enjoy.